for finishing up GI anatomy and function. We now have to follow up after the duodenum. We're basically at a phase now where nutrients have been almost completely digested and ready for absorption, but not quite. And so we'll talk a little bit about the hormones again, but mostly about those segments of the GI tract, just sort of complete the story here. So if we come out of the duodenum, got a gnat attacking me. Right, that's the duodenum, D in there. The next segment of the small intestine, right? So the stomach would be here again, right? And pancreas was here, right? Just for reference points, right? We can even add our pyloric sphincter. And so now we're going to be exiting the duodenum, and at that state, we will have digested a lot of things down to, well, let me back up. Several nutrients are typically taken in as polymers, not always, but typically like proteins. We don't absorb or eat a lot of individual amino acids. We tend to get most of our amino acids by digesting proteins. We tend to get most of our monosaccharides by digesting polysaccharides. Lipids are not a polymer, so they're a little different. We still do digest them. We break down our main form of dietary fats are the glycerolipids, often the triglycerides. So that's glycerol plus 3 fatty acids stuck to it. And again, I'm not really saying much about the nucleic acids just because they're such a small component of dietary caloric intake. So anyway, we're going to enter the second segment of the small intestine, which is much longer, right? The duodenum is about 1.5 feet long. The jejunum is something like 9 or 10 feet long. And so we've got the jejunum. Now what I want to do there is this is where the surface area adaptations take off, right? Here's where we have much more significant plica. Much greater density and numbers of villi and microvilli. So those ridges there showing the surface area adaptations of the lining of the small intestine. And the rationale for that is that we have to have a large surface area for large numbers of membrane transporters, right? We typically have, tight junctions between the cells of the epithelium of the GI lining. Tight junctions serve to limit paracellular between the cell permeability. We don't want to absorb glucose into the body and then have it diffuse back into the GI tract in between the cells because there's an open pathway there. So that open pathway is closed due to the presence of tight junction proteins, claudin and occludin proteins that are found in between the epithelial cells. Okay. Then we can control most of our uptake of nutrients through transcellular pathways, channels, and pumps, and and counter transporters. All right. So jejunum is largely about absorption, but before some of that can occur, what we have to have there. So in other words, we had enzymes secreted by the pancreas and the stomach. The ones active in the duodenum and beyond were secreted by the pancreas, right? They're still active and they're breaking down molecules. There are also enzymes found attached to, or they become attached, probably both, to the membranes of the jejunum. And so what we have are called brush border enzymes. And the brush border enzymes are basically doing the last steps in digestion right before the nutrients are taken up by that epithelial lining. And so the brush border enzymes come in the same variety as the secreted enzymes, and they're doing that processing. Now, so these molecules are transported. Let me draw a villus, right? So the villus is the middle surface area adaptation, right? The plica were the really big ones. The microvilli are on individual cells. I'll draw those too. But a villus looks like this, basically. Right, so these ridges would be the plica. Right? I'd have to draw ridges on those ridges to be the villi. These are the villi. Right, and then on to the next one. Right, and I'm systematic enough. I draw enough lines to make these look like columnar cells. A lot of them. I'm tired. Okay, you get the idea? What I'm going to do is on the surface of those, I'm going to put the microvilli, right? Remember, microvilli are on the individual cells. I'm not going to try and cover this thing with microvilli. But again, just so you get the idea, the microvilli would be attached. Not attached, but part of the membrane, of the apical membrane of those cells. Apical, right? So this is lumen up here. Right? The food content, I keep drawing it as green. Right? And again, it would be broken up into tiny little particles now, but I just keep sort of showing that as a green blob, the food content. Now, we need to get food content, so I'll draw little green blobs, right? And I can draw red blobs and white blobs for the carbohydrates versus the amino acids versus the lipids. For the amino acids and monosaccharides, those are absorbed largely as those monomers, transported across the cells, and what we have in the underlying layer, right? Remember the lamina propria underlies, and has capillaries, underlies the epithelium. And so if I put a capillary sort of winding its way up, and it would be not one capillary, but I'm simplifying, right? And we'd have flow in, and we'd have flow out, off to our to the circulation, but we need to be specific about that because this blood flow from the GI tract is actually going to go to the liver first. That's called the hepatic portal system. I can say a little bit about that. And so our nutrients, let me focus on a green dot here, would be taken up by the cell, transported across the membrane, the apical membrane on the lumen side and the basolateral membrane on the lamina propria side or basement membrane side, however you want to view it, and sort of spit out into the interstitial space in this villus, right? Yeah, this is a villus. Villus, singular for villi, right? So this, let's call it an amino acid, is taken up. Amino acids are very small. It's considered a small solute. Even continuous capillaries, those are the least permeable versions of capillaries, they have the smallest pores, would allow an amino acid or a monosaccharide to pass easily across the membrane. And so that guy would end up in the bloodstream and lots of others, right? And so those nutrients end up going, and they're not, this vessel continues on into other vessels, obviously, but it's going to go to the liver, right? And if you recall, that is the hepatic portal system. Blood flow coming out of the GI tract, venous flow out of the GI tract, does not go into the general circulation. It goes to the liver first through a portal system. That's two capillary beds in series, one after another. First one's here, then through a portal vessel called a hepatic portal vein to the liver, where these nutrients can be taken out of the plasma before that blood hits the general circulation. In other words, before it comes out of the other side of the liver through the hepatic veins and into the vena cava and off into the general circulation. So what the liver is doing, and this hepatic portal system in general is doing, is modulating the blood nutrient levels following a meal. It's sending the nutrients to the liver first to be yanked out of the plasma so that the circulating levels don't go through the roof. Okay, that's good. That avoids hyperglycemia. It avoids hyper amino acidemia, too many amino acids circulating around, right? I say, okay, so that's how many nutrients are absorbed, including, and then we could follow that with water and electrolytes and vitamins, many vitamins, not all, water-soluble vitamins, are picked up and off they go, right? Now, let me make it yellow. We have these and I'm exaggerating the size here so you can see them, size relative to the cells, but we have the bile salt coated fat droplets that we call micelles. Right? And so I can sort of put little blue dots around this as the bile salts. Hopefully you can see these. Right? So this would be an unemulsified. I haven't put the bile salts around that one. That's not emulsified. These two are. This one could glom onto another fat droplet and get bigger and bigger and bigger. Right? Which is, so the blue guys, little blue dots here, hopefully you can see them, are the bile salts. Right? From bile. They're the amphipathic molecules I talked about previously that help solubilize these fat droplets, the micelles. Well, the fats forming a micelle. Okay, how do we absorb this? Right? It's not absorbed like the other molecules. And so what we have is these guys sort of quasi-dock with they're called enterocytes, the cells of the epithelium. And the fats are extracted. There are lipases that are in the lumen here that are acting on the fats there, and those fats can be taken up, right? Not as a micelle, but just the fats are sort of extracted and taken up into these cells. Now the fats are not, right? Fats inside the cell will tend to glom together and form big fat droplets too, so we have to package them again. But now they're packaged and they're protein coated, right? And so if I blow up one of those cells, right? Microvilli. Right? This is irrelevant, not irrelevant, but not associated with this picture other than I'm taking this cell out and blowing it up a little bit, right? So here's my fat droplet. Here are my bile salts. And what's going to happen is the fats are going to be taken up and the bile salts are going to be released, right? So we can sort of start erasing these guys as the lipids are taken up, right? So now the epithelial cell, the enterocyte, is taking up the fats, but it can't just, it has to, right? They're lipophilic. You don't want them stuck in the membrane. You don't want them like drifting back out of the cell. So you got to package them again. So now you're going to protein coat them. So now I'm going to coat them in green dots. I have to make these big enough so you can see them. I'll just do one. Right? Now I've got an emulsified fat droplet. The yellow is the fat. The green now is protein. And this is called a chylomicron. This arrow's not relevant to that. Chylomicron. Now that guy is transported and exocytosed into the interstitium, right? So now I've got this guy, yellow, with green dots, because those are proteins now, not bile salts. The blue were bile salts, now they're green, if you can make that out. And those are proteins, so it's a protein-coated fat droplet called a chylomicron. So I'm going to coat it in green. Sorry, it's the same color as one of my nutrients before. Wasn't thinking about that. Right? Well, that guy, it's protein-coated. Do proteins pass through most capillary membranes? Nope. Right? And these are fenestrated capillaries. They are more permeable, but these are proteins. Proteins generally don't even pass, and it's many proteins wrapped around something even bigger, a fat droplet. It can't enter the bloodstream. How the heck do you absorb your fats? You have, let's see, don't want to make that yellow. I want to make it, running out of colors here, purple. You have a blind-ended vessel called a lymphatic. It's specifically called a lacteal, right? In the GI tract, up into each villus is a spur off of the lymphatic vessel that's called a lacteal. Squeeze that in here. Right? Guess what? lacteal is super permeable, permeable to even a protein-coated fat droplet called a chylomicron. So now I can put that guy, can make it around, not into, but around the blood vessel and into the lacteal, and now it becomes a circulating, well, not, I shouldn't say circulating yet, but in the lymph system at least circulating chylomicron. Green. Protein's still associated with it, otherwise the fats will all glom together. Where does your lymph go? Well, the lymph drains together and together and together into larger and larger ducts and eventually empties into the subclavian veins below your clavicles. And so it ends up in the general circulation. It does not go to the liver first. So if you eat a high-fat meal, majority of the fats are actually absorbed in this fashion and end up going into the general circulation. And then slowly they're pulled out by the liver or adipose tissue, the fats are pulled out. But so your plasma gets very fatty after a fat-laden meal. So if you've ever had that feeling, and I'm not saying this is directly associated, but if you've ever had that feeling like you eat a big fatty meal and you're like, oh, I feel like there's like lead in my veins. Your plasma is no longer clear. It is opaque. It turns yellowish, and I mean not the clear yellow that it normally is, but a sort of, it's not translucent anymore, it's opaque yellow because it's got such a high concentration of chylomicrons in it. So your blood lipids go really high. Now, do they fall over time? Of course they do, right? Because again, they're going to be pulled out of the circulation over time by fat tissue and liver tissue and muscle tissue even to be stored. Okay, so that's how our fats are largely absorbed through the micelle to chylomicron into the lymph, lacteal lymph, and back to plasma, right? Okay, now if we go past, let me get rid of this, right? We've now got our absorption going on, and that continues into a slightly histologically different segment of the tract called the ileum. I'll have it bend around this way. I for ileum. It also has these surface area adaptations. They tend to wane, and this guy's also, 7, 8, 9 feet long, something like that. The surface area adaptations start to wane towards the end. They just start to disappear. And again, largely because we've absorbed all of the nutrients, right? Or most of the nutrients by this point. We've absorbed fat-soluble vitamins, A, D, E, and K with the fats. We've absorbed water-soluble vitamins. We've absorbed water osmotically. It follows the nutrients. So as we absorb all these nutrients, water tends to follow along with it through aquaporins. A little bit of paracellular, but mostly through aquaporins because we have those tight junctions in between cells. So what are we left with, right? Well, we're left with sort of processing the remainder, and we haven't absorbed all of the minerals yet, or even sort of like focused on the mineral absorption yet. And so at the end of the ileum and in the colon, the large intestine, which comes next, it's water absorption, mineral absorption, and I should reverse that. It's water absorption because we absorb minerals. So it's still osmotically absorbed water, but now it's because we're absorbing minerals, specifically not really much nutrients anymore. At the end of the ileum, we find a high concentration, or high density, I should say, of lymphatic tissue, essentially like lymph nodes. Those guys, there's a couple different terms for it. Peyer's patches is sort of an old school name. Now they often call it MALT. Mucosa-associated lymphatic tissue. Sometimes they call it GALT, or gut-associated lymphatic tissue. Again, a lot of different names for the same thing. It's a bunch of lymphatic tissue in the wall of the ileum. And it's not that it stops there. It's probably a little bit, but it starts to wane here in the colon. Why so much lymphatic tissue here? Well, it's because we are what you would consider a hindgut fermenter. This is colon. This is actually the appendix, which actually has a high density of lymphatic tissue. Some yellow there. Why so much lymph tissue? Probably close to half of the cells of your body, I don't want to call it of your body, but associated with your body, are bacteria within the GI tract. Now that's not to say there aren't any any bacteria here. They've just been dramatically minimized by the actions of the acidic stomach and then by the actions of enzymes, especially proteases, along the way. They've killed many of those bacteria. If you've ever had food poisoning, where you introduce way more, and sometimes it's the bacterial toxins they make, but way more bacteria here than the GI tract can actually handle, can kill, and then all of a sudden you have way more bacteria sort of within the gut and, not necessarily attacking the gut lining, but possibly passing through the gut lining. Our body's response is to stop absorbing water and get rid of them basically by flushing out the system. These bacteria are not, most of them are commensal, mutualistic even. In other words, they're not. Most of them are not pathogenic. Most of them are not disease-causing, and that's actually the reason the pathogenic ones don't do so well is because they're outcompeted by the mutualistic ones, or commensal ones at least. Again, so the non-disease-causing ones versus the pathogenic disease-causing ones, they're competing for resources. They do things like make vitamins, vitamin K and some of the B vitamins, which we do absorb, right? They also tend to digest some of the things we can't digest. They digest some types of dietary fiber and release fatty acids, which we can absorb. There are studies into people that have a higher tendency toward obesity. Do they have gut bacteria that are better at, again, there's probably about 1,500 to 3,000 different species here. You are an ecosystem. And so depending upon what those gut bacteria are good at metabolizing, in people where they're really good at metabolizing dietary fat, it's thought that they might actually get more fatty acid absorption. They basically get more calories from their food than other people, tending toward increased obesity in those people, even though they may be eating the same amount as somebody else. Anyway, and so we get vitamins, so K and B, some fatty acids that are being produced, minerals, right? So sodium, chloride, and calcium, and potassium are being absorbed, and because they're being absorbed, we're absorbing more water. And you're left with fecal matter, right, that is largely dry weight, at least, dietary fiber, and dead bacteria, right? There's very little caloric content to it because we've absorbed all of that. Now, again, we can't get the caloric content out of dietary fiber, and so I'm not counting that. So dietary fiber, dead bacteria in fecal matter. All right, are the hormones involved down here? Are they being secreted down here? Some of them. Motilin is secreted along the full length because, again, that's that housekeeping, increased motility in between meals. Some of the others that are active pretty far down the intestinal tract, CCK is active, stimulated to be released pretty far down the tract. A couple others, they tend to die out somewhere in the middle of the small intestine. Very little hormone secretion coming from the colon. All right, I think that does it. Finishes up GI function and the GI hormones. Note that there are a lot of so I hit the 5 main, just to recap, 5 main GI hormones, right? Let's see if I can hit them. Gastrin, secretin, cholecystokinin, GIP, remember that's gastric inhibitory peptide or glucose-dependent insulinotropic peptide. and motilin, right? All five. There are a lot of paracrine agents. Somatostatin is paracrine. VIP is neural, and there's a bunch of others I didn't even mention. Histamine is really important. Histamine, gastrin actually acts through histamine in many ways. So there's a lot of paracrine agents that I didn't say too much about, but that are important players. Right? Again, we're focused largely on the hormones, other than the ones I made a big point about, talking about like somatostatin. Okay.